just a reminder first from, in terms of what we said last week. From the Salk Institute website, we read a couple of excerpts as follows. In the new study, so they're speaking about research that had just been published in, I don't remember, it's like Circulation Research, I think is the name of the journal. In the new study, the researchers created a pseudovirus that was surrounded by the SARS-CoV-2 classic crown of spike proteins, but did not contain any actual virus. Exposure to the pseudovirus resulted in damage to the lungs and arteries of an animal model, proving that the spike protein alone was enough to cause disease. Tissue samples showed inflammation, endothelial cells lining the pulmonary artery walls. And later in that same piece, they said this is the first study to show that the damage occurs when cells are exposed to the spike protein on its own. So since last week, we have been able to read the actual paper. And thank you again to the many of you who forwarded it. And also, I was just able to access it after our stream. As well as a new statement from the authors in which they say that they're clarifying what they think the implications of their work is. As well as a blog post from a researcher in drug discovery who has a regular blog in Science with his take. And we've also noted changes to the Salk Institute's news release, of which there's no indication on the site itself. They don't indicate that they've actually changed.
The changes are present, but it is not acknowledged that there is a change.
Right. And we'll get to that. It's fairly common to change posts, but in this case, the change was distinct enough that if we hadn't actually shown my screen so that it was clear what was up at the time that we were talking about it, it would look like we had been disingenuous or even dishonest in our
it's always possible to miss something, but
not in this case. It was right at the top. So the change is right at the top, and I have screenshots from last week and now to show. And in fact, there's now been an additional change. So, but we'll get there. Again, like I said, published in Circulation Research by a team of researchers, including some at the Salk Institute, titled SARS-CoV-2 Spike Protein Impairs Endothelial Function via Downregulation of ACE2. And it's a short paper. It's a dense paper, very short, very dense, really not sufficient to, fully assess. But I will highlight and read this last sentence, in which they say, this conclusion suggests that vaccinated-generated antibody and/or exogenous antibody against S protein not only protects the host from SARS-CoV-2 infectivity, but also inhibits S protein-imposed endothelial injury. Okay, so thank you, Zach. I also wanted to, before we start talking about that, I will say that from them, which is, again, the last sentence of their paper, in which they seem to say, we don't think this has bad implications for the mRNA and DNA vaccines, which obviously, as we all know, code for the spike protein. But it wasn't clear to me why that sentence was there for, like what it followed from. Here is a statement. So Zach, you can show my screen again, a statement from the authors that came out this week. Which, the third paragraph of which reads as follows. For COVID-19 vaccination, a small amount of the spike protein mRNA packaged in nanoparticles, Pfizer, BioNTech, and Moderna, or spike protein DNA carried by adenovirus, Johnson & Johnson, are delivered into humans through intramuscular injection. The spike protein mRNA or DNA, after translation to modified spike protein, then function as antigens to be recognized by antigen-presenting cells for antibody production. So this is what we've been told. This is consistent with everything we've heard about the vaccines. This is from our live stream last week from the Salk Institute site, in which you can see the top sentence reads, scientists have known for a while that SARS-CoV-2's distinctive spike proteins help the virus infect its host by latching onto healthy cells. Now, a major new study shows that they also play a key role in the disease itself. So if I may, Zach, compare that to what the site looks like as of 2 days ago, in which the second sentence reads, now a major new study shows that the virus spike proteins, parentheses, which behave very differently than those safely encoded by vaccines, also play a key role in the disease itself. So that's been added. There's no reference. And really, I think part of what we're going to talk about here is that this is a claim that is being made by a lot of people. But I don't think that there's any experimental evidence, at least that I've seen, research evidence, that this is actually true, that that the virus spike proteins and the vaccine spike proteins inherently behave differently. We hope they do, and it's suggestive that they might, but we don't actually know this for sure. One more link here, if I can. So this now is the live site as of right now. And they've changed it one more time, which is that they've hot linked that parenthetical which says of the virus spike proteins, they behave very differently than those safely encoded by vaccines. Well, what's the hot link? It's to this same blog post in Science Translational Medicine, which is exactly the same blog post that the authors of the paper in their clarification, in their author statement, point to. So, a couple of things from the blog post, and then I think we've set it up enough that we can start talking about it. So here is a PDF of the blog post, and I'm going to try to start showing the PDFs rather than so that I know that we've captured the moment in time. So this was posted on May 4th, Spike Protein Behavior, by a researcher named Derek Lowe, who has a regular blog in I can't remember now, I think it's Science Translational Medicine, and here are just a couple of quotes from his blog post. He says, so I've been getting questions about what this means for vaccination relative to this spike protein paper out of the Salk Institute. If we're causing people to express spike protein via mRNA or adenovirus vectors, are we damaging them just as if they'd been infected with coronavirus? Fortunately, the answer definitely seems to be no, he says. Later on, now we get to a key difference. When a cell gets the effect of an mRNA nanoparticle or an adenovirus vector, that is the mRNA vaccines versus the DNA vaccines, it of course starts to express the spike protein. But instead of that being assembled into more infectious viral particles, as would happen in a real coronavirus infection, this protein gets moved up to the surface of the cell where it stays. That's where it's presented to the immune system as an abnormal intruding protein on a cell surface. The spike protein is not released to wander freely through the bloodstream by itself because it has a transmembrane anchor region that, as the name implies, leaves it stuck. Later in the same paragraph, the spike protein produced by vaccination is not released in a way that it gets to encounter the ACE2 proteins on the surface of other human cells at all. It is sitting on the surface of muscle and lymphatic cells up in your shoulder, not wandering through your lungs, causing trouble. Some of the vaccine dose is going to make it into the bloodstream, of course, but keep in mind when the mRNA or adenovirus particles do hit cells outside of the liver or the site of injection, they're still causing them to express spike protein anchored on their surfaces, not dumping it into the circulation. So all of that suggests that the researchers and the drug development people believe that the spike protein that is produced by the mRNA and DNA vaccines is both modified slightly and its method of entry into the body both mean that it will be spreading much less widely than an organic encounter with SARS-CoV-2 in the wild, such that actually this research suggests that we should feel better about these vaccines than we did before this research. That is the conclusion here, and I think it's plausible, but it's not the only interpretation.
Well, let's just say a couple things are going on. One, that the idea of the protein having an anchor that will keep it in the cell surface is the idealized version of what's going to go on. So kudos for designing it in this way. And let me put that in the context of how the vaccine should work under circumstances where everything goes as planned. So let's take the mRNA vaccine, one of them. You've got this lipid nanoparticle coat. It's covering an mRNA. It gets into the body through injection. It encounters some cells for reasons that I think are still a bit murky. It gets taken up. That could be as simple as the nanoparticle coat having an affinity for the membrane of cells that causes it to be taken up. Big asterisk there. Which cells take it up? Why do they take it up? How different is it? How many cells take it up in one patient versus another? These are all big questions. Maybe somebody knows the answer, but it's not easy to find even for somebody who knows that they're looking for it. So you've got the mRNA gets dumped into a cell. The cell, because all cells in the body make proteins from mRNA messages that are usually coming from the nucleus of the cell, just simply translates this thing automatically. And the nature of the protein that gets translated has this anchor bit in it that, if all goes according to plan, results in it sticking off the surface of the cell, pointing out. At which point, the hope is it will be encountered by antigen-presenting cells, which will detect this cell, will effectively take it up, and then present it to a bunch of naive T cells, that fraction, that tiny fraction of the T cells that it encounters will react to it because they are wired in such a way that they have an affinity, a low affinity at that point. They will be triggered to diversify then, and selection will create an ever better affinity for that spike protein.
The short version of that being, the hope is these cells with the spike protein having been produced that are on the surface of them will be recognized by our immune system, and thus we will be able, our adaptive immunity will go into effect and produce a response such that should the person who is thus vaccinated encounter wild type SARS-CoV-2, their immune system will then be primed and ready.
Right. It will not be naive, even though they haven't encountered the virus before.
Right.
And I should just say that in some sense, what this is a benevolent hijacking of the immune system, because what the immune system is doing, these antigen-presenting cells are maneuvering around the body, looking for our cells that have been infected. So if your cell has been infected by a virus, it expresses some proteins that are native to it, and it expresses some proteins that are novel. And so these cells, by finding our own cells that are displaying novel proteins, get an indication of an infection. And then they take the evidence of that infection to a place where cells that are capable of responding to the infection are lurking, the lymph nodes, for example. And anyway, it accelerates the process by which your immune system discovers the enemy, gets a description of it, and then figures out how to recognize it really well and creates an army of cells that can then fight it. So what we do is, in this case, we trigger the presentation of this antigen on the surface of a cell that is not infected.
Being what?
Humanity, those who are deploying the vaccine.
We who are vaccinated.
Well, scientists are using...
I'm trying to figure out what we is here.
We, the science technology complex, are triggering human cells in an uninfected person to display an antigen as if it was infected. Then the antigen presenting cells take over doing their normal job, collect that antigen, display it to cells that are capable of learning the formula and responding, and the whole thing unfolds in this way. The problem with this story, as deployed in that blog post, is that it assumes that everything works as we hope it will. And there's actually very little chance that that's right, because what we are doing is completely novel with respect to the body, right? We are taking a pseudovirus, right? A lipid nanoparticle coat around an mRNA. We are infecting a cell with an mRNA that encodes this one protein, hoping to get it displayed on the surface. But none of this says that that's what happens. It certainly is happening enough that these vaccines do create immunity. That is clear. But it does not tell you how much of the spike protein is actually going through this process in the way the textbooks suggest, and how much of it is ending up somewhere else. So for example, basically what I see in this blog post and all of the analyses that point in this direction is a cherry picking of the information. Here are all the reasons that this is probably safe, and here are none of the reasons that it probably isn't, right? And the point is, you've got a collection of both reasons. They are correct. If the protein anchors in the surface of the cell and the antigen collecting cells find it and they take it to some T cells that get a chance to learn it, then you're not going to have the effect that these authors found when they dumped spike protein into the lungs of what turn out to have been Syrian hamsters.